Hi there. We'll get started with the webinar. So hi everyone, my name is Irina and welcome to the April edition of Research Reels. So we'll do our little spiel to begin this webinar just about IMPART. So who is IMPART? IMPART is a national research team headquartered at Dalhousie University, New Brunswick. But although our headquarters are at Dalhousie, we do encompass many universities and clinical partners all across Canada. So we have 45 academic partners, 85 universities, or the words are just getting jumbled. And Research Reels is something that we created a little while ago. It's a monthly seminar series that we use to host to improve networking across IMPART members. And this is aimed to enhance opportunities for future collaborations and support translations of research findings. So we're hoping by opening up the floor to research that we'll have our IMPART research members collaborate with each other through this. If you enjoy these webinars and you want to nominate a speaker, stay tuned for a post-webinar survey where you can do that there, as well as provide us with some feedback on these webinars. IMPART is present across all these different social media platforms. So as you can see, we have Facebook, Instagram, LinkedIn. So feel free to follow us there to keep up with some of our new research and with our members. We have all our members listed on impart.team, which is our website, if you want to learn more about each individual member's research. And as well, these research reels are posted on our YouTube channel. So that's Research IMPART. As you can see right here, we have all our seminars here if you want to check some of the past videos out. So now let's move into introducing our speaker for today's webinar, who is Dr. Matthew Stoyek. So Dr. Matthew Stoyek is a postdoctoral fellow in the Department of Physiology and Biophysics at Dalhousie University. He completed his PhD in the Department of Medical Neuroscience before moving on to his current position. His work focuses on regulation of cardiac output, particularly by the autonomic nervous system. Dr. Stoyek uses electrophysiology, optogenetics, immunohistochemistry, pharmacology, and imaging techniques to perform integrated physiological investigations of the functional roles of cardiac neurons in regulatory pathways. He has established the zebrafish as a model for studying intracardiac nervous system control of pacemaker function and is currently focused on using genetically modified zebrafish as a model for investigations of human cardiac autonomic pathophysiology. So without further ado, I'll pass on the floor to Dr. Stoyek, who will teach us how to fish.
So thank you for the introduction, the kind introduction, and thank you all for being here today. As was just said, most of my work is looking at neural regulation in the zebrafish. However, I'm hoping to highlight a few things overall, including some other findings looking at cardiac function in the zebrafish, just to highlight how I see this little fish becoming a big model in cardiac research. So I'll do this really quickly. So to start off at its most basic, your heart is quite simply an electrical pump. And over the course of your lifetime, it will contract 2 to 3 billion times. The interesting part of this is that no two beats ever happen in the exact same way. And this occurs without you ever thinking about it or ever having to do anything for it. An example of this is when you begin to exercise. Almost immediately, your body senses that you're exercising, that activity levels have increased, and will adjust to increase your heart rate, increase cardiac output to meet the new demands for oxygen and nutrients that your muscles now have. Then once you're done exercising and your body then senses that, and all of a sudden your heart rate slows, cardiac output decreases, and you reach your kind of normal resting level of cardiac activity. Now, as was said, I look at nervous system control, and this is the primary extrinsic or component outside of the heart that controls the cardiac function. And admittedly, this is a very complex slide, but all I want you to take from this is that control of your heart's function by the nervous system depends largely on a very constant and complex interplay of neurons in the central nervous system in the brain, neurons associated with the spinal cord, and neurons within the heart itself. And the interplay occurs through a constant communication between all of these levels of neurons, all of these neurons within your body, to maintain this optimal level of supply of oxygen and nutrients through the blood to your muscles and other tissues of your body. Now, given the importance of this control by the nervous system, it has been studied quite in depth. And in fact, the first publication on innervation of the heart was actually printed in 1794. So we've had more than 200 years of knowing that there's a role for the nervous system in control of the heart. However, even to date, a lot of this research has depended upon the use of mammalian models. Now, in a lot of ways, this is a pro because their hearts are very similar to our own, and findings from these studies become very translatable to humans and to patients. However, the very tissue-dense nature and often size of the mammalian heart poses a considerable limitation on some of these studies, especially when one takes into account that the nervous system, as is represented by these arrows that travel all along the heart here, the nervous system actually can have effects locally or project at great distances to where the neurons originate from. And as I said, with the tissue dense nature of the mammalian heart, a lot of researchers, even in current and recent works, rely on reduced preparations of the heart, meaning they'll take, say, a chunk of the atrium or a piece of the ventricle that contains the neurons of interest. However, in doing this, we get insights into the function and specific functions of these neurons. However, we're limiting what we conclude about global or whole heart function of these neurons, because what we're doing is then severing these projections to more distal areas of the heart. And we can get an idea of what's happening locally or very specific functions of those neurons, but we don't know what it means on a whole heart level. Now, about the time that I was starting my PhD, the zebrafish had become a very powerful model in cardiac research, owing to its ability to regenerate its heart tissue, often without the presence of scar tissue. And as you can see here, this was one of the first publications, dates back to 2006. And even more recently, they've shown that extract from the zebrafish heart can actually induce regeneration in mammalian mature hearts, which had previously been thought to not be able to regenerate. So as you can imagine, there's been a lot of work in terms of molecular biology and genetics going into these studies. And from that, we knew that a lot of the genetics and genes expressed, proteins expressed within the zebrafish heart could also be found in the human heart. However, at that time, very little to almost nothing was known about the function of the heart. We had crude insights into heart rate and different performance indices. However, overall function and control was largely unexplored. This was a little bit surprising, given one of the biggest benefits of the zebrafish is just how easy it is to image and visualize the whole heart, as well as the fact that while the zebrafish heart has a very different anatomy, it's a two-chambered heart. You can see a single ventricle and a single atrium. This is essentially a human heart split in half and serves the same physiologic function. Nobody had looked into this in any real depth. And as I say, you can visualize using standard microscopes. Both of these images were taken with microscopes that are found in many labs at many universities. And you can see the whole heart in the adult just simply by opening up the chest. And even more interesting is the fact that within the juvenile zebrafish, you can see the heart in this area here in a whole animal. And taking that one step further, in our lab, we've now started using zebrafish, genetically modified zebrafish, which express fluorescence proteins, in this case, a green fluorescence protein, within the muscle of the heart, which allows us to observe both anatomy and function in a whole animal using standard light microscopes. Further to this, as you can see here, this is an immunohistochemistry image. Is in green is the nervous tissue within a maturing zebrafish. In white is the vasculature, and in red is the heart. But you can see in great detail using standard confocal microscopy, very great detail of what is actually happening in the nervous system and the heart in a whole animal. And what I want to point to draw your attention here to and point out is this cluster of neurons at the back of the brain, which forms the vagus nerve. And in zebrafish and humans, this is cranial nerve 10, and what provides the majority of the neural input from the nervous system to the heart. And in this case, you can see this nerve tracing all the way down from the brain through the body to the red tissue of the heart. And maybe if your monitor is big enough, that you can see actual projections going through the walls of both the atrium and ventricle from the vagus nerve. Again, just highlighting how easy it is to visualize the entire system in this. And this is something that's still to date not possible in any mammalian model. So in the sake of time, I won't go into great detail here, but a lot of my work during my PhD focused on establishing that the zebrafish heart contained all of the components of mammalian cardiac regulation. And the take home here is that it does. That in panel A, what you can see is that the neurons are in black, projecting around in this case, the atrium of the human heart. In panel B is a Mohs heart, and in red, you can see the nerves traveling throughout the atrium at top and the ventricle. And then most of greatest interest to me, at least, was in panel C in the zebrafish heart. In white, you can see innervation throughout both chambers of the heart. And it's a very robust innervation. There's axons and projections of these nerves throughout both chambers into the aorta. And as I said, everything that we looked for from the human heart in the zebrafish heart, we could find. So at least anatomically and based on this immunohistochemical data, we had suggestion that we had a good model, that the zebrafish could be a good model for studying the cardiac innervation. Now that said, we had anatomical data, and the next step then was to look and see if this functioned in the same way. And the first step was then to isolate a heart from a zebrafish and put it into a dish and just make sure it stayed alive. And here you can see within a temperature controlled bath that this heart from a zebrafish just kept happily beating. And in some cases, I've had them in this dish for up to 12 hours. So we then had a working preparation. In this bottom panel, it's just a kind of a higher magnification of the sinoatrial region. This is the area of the atrium from above. These two white lines are the vagal nerves coming in, and down here you can see an electrode. And when I start this film, what you'll see is that the heart beats in normal sinus rhythm, as is shown by this ECG taken from these leads. Then when this red dot comes on, the heart slows almost immediately. And what's occurring there is that I'm delivering an electric pulse to the nerve, simulating central nervous input. And this causes this immediate bradycardia during a period of nerve stimulation, and followed by a recovery tachycardia. Again, very reminiscent of what is known to occur in mammalian models and in humans. So again, as with the neural components, we wanted to ensure that this was happening through the same pathways as in humans and mammals. And in humans, neurons from the brain send projections out and release neurotransmitters onto neurons within the heart. This activates the cardiac neurons, which release neurotransmitter onto the tissues of the heart, causing changes in function. Now, when I was doing in the last video where I was showing the slowing of the heart through vagal stimulation, I'm applying a stimulus here. So I'm activating release, in theory, activating release of transmitter, which activates the whole cascade to the tissue of the heart. This is shown a little differently here when we quantify it. This is shown through changes in our interval, or measuring the distance between the spikes on an ECG. And the longer the distance between spikes, the lower the overall heart rate. But when I activate the vagal nerve, we see this increase in interval, our interval, which again is a decrease in heart rate. When this is then, we apply a blocker to the receptors on this neuron, we see an ablation of this, suggesting we are getting transmission from one neuron to the next, causing this effect. Finally, if I apply a blocker to the receptors on the tissue of the heart itself, we again see this ablation. And for us, this was acted as support or confirmation almost that the pathways are acting through the same neural cascade that we know exists in humans, and supporting the idea that we do have a good model in the zebrafish to begin to investigate cardiac function and neural control. So Serena mentioned, I look predominantly at control of the pacemaker. This is what drives the automaticity of the heart, or what drives constant contraction and the beating of a heart. And in humans and in mammals, it is known that the process of excitation and contraction from the pacemaker occurs through cycling of voltage and calcium levels within the myocytes of the heart. And in this panel, you can see an isolated zebrafish heart, atrium on the bottom, ventricle at the top. And the following panels in red is this heart. These are both taken simultaneously onto two cameras. So the activity you're seeing is occurring at the same time. In red is a voltage sensitive dye that's been loaded into the heart, and in green is a calcium sensitive dye. And both of these increase in fluorescence when when voltage increases or when calcium levels increase in the heart. And what I want you to see in this video, when it plays and when it loops back to the beginning, is that you can see both electrical and calcium activity, but this activity initiates in the sinoatrial region. The electrical activity initiates within the sinoatrial region, travels out to the ventricle. Now, when we look at the calcium, the voltage happens first, but this is very rapidly followed by a calcium increase, suggesting to us that this calcium voltage cycling that occurs in human hearts is also occurring in the zebrafish heart. The next step then for us is to look at the cellular mechanisms, the most basic mechanisms of this, and to see, are they similar to or the same as what we see in mammals. And what you can see here is just a schematic of the zebrafish heart, and the panels that I'll be explaining here are within this area of the atrium. First thing I want to show is this ring here of very distinct tissue, and this is the sinoatrial node in a zebrafish, and it fully encompasses the area surrounding the sinoatrial valves at the in the atrium. And the cells within this region just exhibit a very distinct spindle-shaped morphology, which is very stereotypic of what is observed in both humans and mammalian models. So suggesting that we do have distinct sinoatrial node cells. Now, as I said in the last slide, and we saw from that video, is that it's calcium and voltage cycling that drives excitation and contraction of the heart. And in the human heart, the main contributor to this voltage component, or voltage clock as it's called, is the HCN4 channel, which is driving what's called the funny current. And when we look within the sinoatrial region, what we can see is that there is an increased expression of HCN4 in the nodal tissue, almost little to none within the tissue of the working atrial wall. Then when we look at calcium handling channels, in this case, this is the ryanodine receptor, we see that it too has high expression within the sinoatrial node cells, but it is expressed at a lower level throughout the working myocardium. And again, this is expected from what we know of mammalian cells. So we know at least the basic mechanisms are present and distributed in the same way that they are in mammals and humans, but are they acting through the same mechanisms? And to do that, what we did was applied blockers of either the membrane clock channels or the calcium clock channels. So these are the channels that cycle voltage and calcium within the pacemaker. And what we saw was that, for the most part, the blockers that are known to work in humans to block either the funny current through HCN4 or calcium through a number of channels cause the same decrease in heart rate in zebrafish that they do in humans, supporting our theory that the pacemaker is driven through the same mechanisms in zebrafish, and adding support to the zebrafish as a model, saying that not only are we studying neural mechanisms that work through the same pathway, they're controlling cellular mechanisms that also work through the same pathways as in humans. So this then gave us a model to begin to look at, for lack of a term, pathological conditions, because understanding neural physiology is interesting, but a lot of us work in medical, in the medical faculty, and want to know, what does this mean for pathology? So the first condition that I looked at was aging. And what you see in panel A is a very young zebrafish versus panel B, a heart of a very old zebrafish. And at least superficially, the innervation is shown here in green, looks the same in both the very young and the very old zebrafish. However, when we looked at this in more detail, we began to see that the number of neurons shown in this kind of pink magenta color here actually continually increase all the way from 3 months until 12 months, and perhaps even beyond, which is a condition that's very different than the human heart, where aging is known to cause a loss of neurons in some cases. However, when we looked at the function of the nervous system in these hearts, we saw that activating the vagal nerve at a very steady, consistent rate through all these age groups caused a significantly larger decrease in heart rate in the young animals than it did in the old animals, suggesting that although the number of neurons was actively increasing as the animal aged, their overall function was for some reason decreasing. And the exact mechanisms and reasons behind this we're still looking into, but this again is a condition that is known to occur in humans, supporting our use of the zebrafish as a model for investigation of cardiac regulation. We then went on to actually model a human disease, a human pathology. And in our case, when I started during my postdoc, the lab already had a zebrafish which recapitulated a mutation in the POPD-C1 gene. And this gene codes for a protein which is involved in cyclic AMP handling, and had been identified in a population of patients in Europe, which exhibited both muscular dystrophy, but also stress-induced cardiac arrhythmias. And what I mean by stress-induced cardiac arrhythmias is that they were more prone to developing arrhythmia during periods of exercise or intense activation of the cardiac neural pathways. So we had this fish on hand to looking at electrical function within the sinoatrial node, but it was also known that while POPDC was expressed in the myocytes, cardiac myocytes, and this is likely what contributed to some of the electrical dysfunctions we were seeing, and as well as it was being expressed in skeletal muscle, which explains the muscular dystrophy. It had been described very, very briefly that the neurons within certain regions of the brain very highly expressed POPDC1. Now, given that we had this component of a stress-induced cardiac arrhythmia, and as I said, this is typically involved with activation of at least one branch, the sympathetic branch of your nervous system, I wondered if, at this point, if this expression of POPDC1 within neurons also occurred within the neurons of the heart and was contributing to these arrhythmias. So our first step in this was to look at the hearts of the POPDC1 mutants. And while all of these animals are homozygous carriers for the mutation, meaning they carry 2 copies of the mutation, we did observe a drastic range of phenotypes within the innervation of the heart, ranging from this very normal looking innervation in panel D, which had all of the neurons that were to be expected within the heart of the zebrafish, through this moderate phenotype shown in panels E, which again, you can see a reduction in the innervation of the ventricle, a reduction in the innervation of the atrium, kind of beginning appearances of disorganization of innervation throughout the chambers. And typically these animals only had about 75% of the total number of intracardiac neurons that were present in a normal animal. Finally, we had what I've labeled as the most severe, which, as you can see here, had to have a very disorganized cardiac innervation and a very severe reduction in the total number of neurons. In most cases, I've said in this image about 50% or less than 50%. In a lot of cases, they only had 20 to 30% of the total number of neurons that a normal zebrafish would have present in the heart. So my next question then was to say, what does this mean functionally for the animal, knowing that the patients are more prone to these stress-induced arrhythmias? And what I found was when I tested these animals, just using the ECG system that I showed in the previous slide, we had normal rhythm, and they were then exposed to a vagal nerve stimulation, which slowed the heart rate, and again, rebound tachycardia, perfectly what we had expected from previous experiments. And when we looked at these mutants that performed well under these stress tests by nerve stimulation, we found the nervous system in the heart was by and large perfectly normal. However, in the subset of animals that displayed the most severe arrhythmic susceptibility, as you can see here, the vagal nerve stimulation, perfectly fine response throughout, but then devolves into a period of fibrillation-like activity. When these animals' hearts were investigated after these tests, again, this heart is actually from this ECG, saw this very distinct correlation of poor cardiac performance being linked to these cardiac innervation deficits, and suggesting that there is likely a role for these deficits and defects in the progression of the disease associated with POPDC1, at least in terms of arrhythmogenesis. Then, almost through accident, well, essentially very much through accident, in delivering some of these vagal simulations and observing the animal devolve into arrhythmogenesis, or into a rhythmic period, sorry, I found that reactivating the cardiac nervous system at what I'm terming a subthreshold level, and what I mean by subthreshold here is that I'm delivering electric impulses to the nerve, stimulating the nerve, but at a level that is subthreshold for heart rate effects. So this is very likely activating only a very small subset of cardiac neurons. But what you can see here is that as soon as this subthreshold nerve stimulation is delivered, almost immediately snaps the heart back into a normal sinus rhythm. And this not only worked within animals that I put into arrhythmia through vagal nerve stimulation, but animals that developed spontaneous arrhythmias could also be snapped back into normal sinus rhythm very quickly by delivering this same subthreshold nerve stimulus. And what we think is happening here is that we're restoring, given the loss of neurons that we'd seen that I talked about in the previous slide, we're seeing this reduction in activity, again, also shown with the aging. And by delivering this subthreshold nerve stimulation, we're restoring some of the activity that may have been lost. And this is actually kind of very hotly debated right now in terms of human clinical trials, that vagal nerve stimulation can be both pro-arrhythmogenic and alleviate a lot of arrhythmic symptoms depending on the study you read, but it's beginning to be viewed as a very promising potential therapeutic for people with arrhythmias. Now, in this case, as I said, I'm electrically activating the vagal nerves, which is a little bit messy because there's multiple nerves in there. I'm activating multiple pathways. And I wanted to then do this, but in a much more controlled manner. And to do that, and the last thing I'll talk about is some work that we've been doing using optogenetics. And for those people who may not be familiar with it, optogenetics is simply using genetically modified zebrafish, which express channels in the tissue of interest, which are activated by certain wavelengths of light. In our case, it's a blue light that activates them, and essentially the light is then acting as to increase activity in the neurons in place of the electrical impulses that I had been delivering previously. And the A, B, and C panels are three animals that we are developing here in the lab. Panel A is an animal which expresses this channel rhodopsin in all of the neurons, and it's very specifically expressed in neurons only. And panel B is looking at an animal, this is the hindbrain of an animal which expresses the channel rhodopsin only in parasympathetic neurons. And in panel C, an animal which expresses only in sympathetic neurons. And what I'm hoping to do with this is create a situation where I can more specific tune activity of these neurons by flashing light onto them and activating them. And what you can see here is just a proof of concept experiment that I did. This is an optical measure of heart rate. You can see the spikes associated with the heart rate. And when I turned light on, in this case, I was illuminating the brainstem. It caused this decrease in heart rate, which at the termination of illumination, heart rate went back to normal. So this will then allow us to very specifically target populations and subpopulations of neurons, both in the brain and the heart, as potential therapeutic targets without the messiness of bulk electrical activation. And this is something we're still actively developing these lines and actively working on this. So this is a stay tuned for further results slide. And with that, I just want to thank a lot of different people over the years who have been involved with this. In my current lab with Dr. Quinn and my former PhD supervisors here at Dalhousie, as well as Dr. Brand and Imperial College, who provided the transgenic POP-DC1 animals, and hopefully have some time for questions.
Thank you so much for that presentation. Your slides are beautiful. I just want to point that out.
Thank you.
So I will stop recording and we'll jump into the Q&A portion.